Hello everyone, welcome you all in our YouTube channel Vaishnavi Nalawade. I am working as a lecturer in Indian Institute of Pharmacy, Sadavli. In today's video lecture, we are going to learn our basics of phytochemistry chapter. In that, part 4. So phytochemistry means the phytochemistry is the study of phytochemicals, which are chemicals derived from plants. Okay, that first one is microwave assisted extraction. Microwave assisted extraction is a conventional technique for the extraction of active components from medicinal plant using microwave energy to heat solvents containing samples, thereby partitioning analytes from a sample matrix into the solvent. So you can see this image of microwave assisted extractor. Okay. Microwave assisted extraction, in that microwaves, that is frequency between 300 megahertz to 300 gigahertz, heat up the molecules by dual mechanism of ionic induction and dipole rotation. The two types of oscillating perpendicular fields that generate microwaves are the electric field and magnetic field. Both the ionic conduction and dipole rotation are responsible for heating of substances. At a frequency of 2450 MHz, the process of heating occurs. Microwave assisted extraction process, in that penetration of the solvent into the solid matrix, solubilization and breakdown of components, transport of the solute out of the solid matrix, migration of the extracted solute from the external surface of the solid into the bulk solution, movement of the extract with respect to the solid, then separation and discharge of the extract and solid. These are the process of microwave assisted extraction. Components of instruments used for microwave assisted extraction, in that magnetron is microwave generator that generates microwave, then wave guide that is directs the waves from source to cavity, then applicator, in that sample holder and sample are placed, then calculator, in that regulates the movements of microwave in forward direction. These are the components of instruments of microwave assisted extractor. Advantages of this method, in that microwave assisted extraction is a much potential substitute to the traditional solid liquid extraction method due to the following advantages over Soxhlet. आपण Soxhlet apparatus यूज काय advantage ते आपण बघितलेला आहे. So in this lecture we will discuss about the microwave assisted extractor. So in that it is less time consuming, as it takes only a few seconds or to few minutes, that is 15 to 20 minutes to complete the extraction process. It requires less solvent, only a few milliliters. It improves the extraction yield. Then since it is an automatic process, it provides better accuracy and precision. It can be used for the extraction of thermolabile constituents. It can be used for extracting minute traces of constituents including heavy metals and pesticide residues from a few milligrams of plant sample. It provides agitation during extraction, which improves the phenomenon of mass transfer. Since its instrumental setup, like Soxwave, combines the features of Soxhlet as well as benefits of microwave, it makes extraction even more attractive. These are the advantages of this microwave assisted extraction technique in phytochemistry. Then techniques used in the isolation, purification and identification of crude drugs. In that, the progress in the isolation and analysis techniques has led to the identification of many unknown compounds. For isolating a particular compound from its plant material, various processes are involved. So these isolation processes depend on the nature of the active constituents present in the crude drug. For example, trapping of the components is done for volatile chemicals, while extraction of non-volatile compounds is done using organic solvents. Isolation of components is done for both known and unknown constituents, and these demands various processes for the separation, purification and identification of compounds along with biological screening. When the required crude extract has been isolated from the plant, the marker component needs to be isolated and identified for its study with respect to chemical nature or for the development of newer formulations. The advances in the field of chromatography have allowed the separation and purification of compounds. After isolation, identification and purification of plant constituents, the class of the compound and the particular substance within that class should also be determined. Then complete identification depends on measuring the other properties such as melting point, boiling point, optical rotation, and retention factor, and comparing the data with those in standard literature with the technique of ultraviolet, infrared, nuclear magnetic resonance and mass spectral measurements. For the final confirmation of known plant compound that has been identified on the above basis is compared with the standard sample, while the identity of new compounds is confirmed by chemical degradation or by the synthesis of compound in laboratory. So these are the techniques used in the isolation, purification and identification of crude drugs. Then next one content, that is spectroscopy. So spectroscopy involves the study of interaction of electromagnetic radiation with matter. So various spectroscopic methods that are utilized for identification of natural compounds, in that ultraviolet, IR, NMR, these are the examples of these identification techniques. And that first one is ultraviolet, that is UV and visible spectroscopy. Ultraviolet and visible absorption techniques involve the analytical methods which measure the light absorption line in the wavelength region from 190 to 900 nanometer by different substances. So the wavelength region from 190 to 380 nanometer is the UV region, and that from 380 to 900 nanometer is the visible range region for the electromagnetic spectrum. So absorption in the UV visible region results from the electronic transitions within the molecule. So many instruments are available for measuring light absorption in the UV visible region of the spectrum. In a single beam instrument, light passing through a monochromator enters the sample, and then the detector in a dual beam instrument and then detector. The next one is dual beam instrument, in that the ratio of the intensity of beam coming through the sample cell to a second beam which does not pass through the sample is measured. Then multi-channel detectors are used in the double beam instrument. Then the separation step and absorption measurement in UV visible region is the most widely used analytical procedure in the field of pharmacy. So the UV spectrum along with other analytical data often serves as a tool for identity confirmation and is also used as detector in HPLC. So the absorption spectrum spectra of every plant constituent are different in specific range of visible light specimen. Every dilute solution should be used and the wavelength should be recorded in nanometers with maximum and minimum of the peak. So in this, these are the spectral ranges of some important group of plant constituents. In that, anthocyanin, beta-cyanins, carotenoids, anthraquinone and its derivatives, and flavonoids, chlorophyls. These are the UV range and visible range. These are the spectral ranges of these plant constituents. Then IR, in that infrared spectroscopy. The IR spectroscopy is used for the identification of many functional groups present in the compound, as different functional groups have specific absorbance in the form of radiant energy in the electromagnetic spectrum. The IR spectroscopy can be measured on spectrophotometer. So the IR spectrum is divided in the following three regions. In that, near region ranging from 1250 to 4000 centimeter inverse. Then mid region ranging from 4000 to 400 centimeter inverse, and far region ranging from 400 to 20 centimeter inverse. Okay, these are the three regions in IR spectrum: near, middle and far region. So the IR spectrum can be measured by the following two methods: in the form of chloroform or carbon tetrachloride solution, and in the form of potassium bromide pellets. The IR spectroscopy is being widely used for identification of drugs, polymorphic modifications, and excipients and raw materials used in the pharmaceutical manufacturing. This is because it is highly sensitive and the spectra can be easily obtained on any type of sample, including insoluble solids, polymers, solutions, and gases. Obtaining superimposable spectra, that is identical IR spectra of two samples, proves that the two samples are similar in their chemical structure. The IR spectrometric analysis helps in the structural elucidation of functional groups of biomolecules. It also aids in the quantitative analysis of antibiotics, alkaloids, quinine and strychnine. It is 6.2 and 6.06 micron. So steroidal sapogenins 11.11 U, etc. So these are the IR frequencies of some important groups of plant constituents. In that, alkanes characteristics bands. Approximately, these are the values of alkanes, alkenes, acetylenes, alcohols, phenols, aldehydes, ketones, esters, carboxylic acids, and amines. The infrared spectroscopy is also used for fingerprinting analysis for identifying the compounds qualitatively. The next one is nuclear magnetic resonance spectroscopy, that is NMR. So the NMR spectroscopy involves absorption of radioactive constituents at limited radio frequency in a particular magnetic field, which differs from compound to compound. So there are two types in NMR techniques, in that proton NMR and second one is carbon spectroscopy. In proton NMR, that is one hydrogen NMR spectroscopy. So this type of NMR spectroscopy measures the magnetic moments of different hydrogen atoms. This helps in determining the number of hydrogen atoms in a particular compound and or a particular group, thus establishing the compound structure. So 13 carbon, second one, that is 13 carbon NMR spectroscopy. This type of NMR spectroscopy measures the nature of carbon skeleton of a particular compound. For NMR analysis, the compound is dissolved in a standard inner solvent known as TMS, that is trimethylsilane, which is subjected to a very strong magnetic field. The different types of proton, depending on their molecular nature, undergo various chemical shifts, which are represented by delta. So the NMR is used as an important tool in the elucidation of molecular structure, especially the stereochemistry and configuration. It determines the position of protons in a complex molecule, and it is used for the identification test in pharmaceutical analysis. It is also used in quantitative analysis in the sense that a reference standard is not required for the substance being analyzed. NMR is widely used for determining impurities and minor components in the mixture due to its easy speed and specificity. So NMR can also be coupled with mass spectrometer. These are all about NMR spectroscopy. Then second one is mass spectroscopy. The technique of mass spectroscopy measures the mass to charge ratio and abundance of gas phase ions to identify the amount and type of chemicals a sample contains. So the information recorded using a small amounts of sample and contained in a single mass spectrum helps in the identification or structural elucidation of an unknown compound. In many newly developed ionization techniques, there is no need to vaporize the sample prior to ionization. This has made various compounds of biomedical and biochemical importance responsive to mass spectrometry. A gas chromatograph is used as the mass spectrometer inlet to enhance the high sensitivity and specificity of the mass spectrometer and combined gas chromatography mass spectrometry. GCMS is currently a powerful analytical technique. Even the highly sensitive immunological techniques cannot offer the high sensitivity and specificity of GCMS. Various major developments have been made recently in GCMS methodology, and these include the direct coupling of capillary gas chromatographic columns to mass spectrometers, the availability of ion source for chemical ionization, and the increasing use of isotopically labeled internal standards in assays using quantitative selected ion monitoring, that is QSIM. MS relies on the basic principle of utilizing an appropriate method to generate ions either from inorganic or organic compounds, then separating these ions on the basis of their mass to charge ratio M by Z, and finally detecting them qualitatively and quantitatively by their respective M by Z, that is mass to charge ratio and abundance of gas phase ions. So the analyte may be thermally ionized by electric fields or by impacting energetic electrons, ions or photons. The ions can be single ionized atoms, clusters, molecules or their fragments. Static or dynamic electric or magnetic field affects the ion separation. The mass spectrum obtained is a 2D representation of signals intensity on the ordinate versus M by Z on the basis of abscissa. Then fluorescent analysis, next one techniques. In that different compounds show different fluorescence in different organic solvent because the light absorption through compound molecules takes place in a definite wavelength range of the light. As a result, different radiations are re-emitted from the molecules and different colors are seen in the UV light. Many natural products that do not visibly fluorescence in daylight produce fluorescence using UV light obtained from mercury vapor lamp or tungsten. The fluorescent lamps are fitted with a filter which removes visible radiation from the lamp and transmits UV radiation of definite wavelength. For example, cinchona bark shows many luminous yellow and few light blue patches under fluorescent light. The other qualitatively evaluated drugs are Columba shows yellow fluorescence, but the phloem and cambium show dark green fluorescence. So, hydrastis rhizome show golden yellow fluorescence. Rheum imodi shows brown fluorescence. Rheum rhaphnioticum shows violet fluorescence, ergot powder shows red fluorescence, olive oil shows deep golden yellow fluorescence, arachis and sesame oil shows blue fluorescence, berberry shows yellow fluorescence, ginseng and aconitin shows light blue fluorescence, imitin shows orange fluorescence, precipitate chalk that is shows gray is violet fluorescence. These are the fluorescent analysis which helped in the identification of this drug. The result components can be located on the developed TLC or paper chromatogram by observing under UV lamp or by Gcms method on TLC plates. So quantitative fluorescence analysis using the fluorescence produced by a compound in UV light can be done with fluorometer or spectrophlorimetry. So for example, senosize extracts of rhubarb and berberries and cinchona alkaloids can be evaluated using this technique. So fluorescent analysis of different constituents, that is terpenoids, anthraquinone, tropane and indole alkaloids. These are the solvents used in the fluorescent analysis and color of fluorescence. So with this we are conclude the fourth part of this chapter. So the remaining content will be discussed in next video lecture. Thank you. Happy learning. Stay safe. Keep studying more, learning more and practicing more to understand the lessons completely. And please subscribe, like and share more and more to my YouTube channel. Thank you.